Hello everyone, welcome back for some more biotechnology. So as we mentioned in the previous video, we are going to spend the bulk of our remaining time in this chapter discussing chromatography, which is going to form the basis for how we do most of our protein purification. So this video is going to be a relatively short one, but the next video is going to discuss in depth the different types of column chromatography that we can use to separate and purify proteins on the basis of their many different physical properties. So let's go ahead and start by introducing ourselves to the actual principles behind how chromatography works. So first of all, what is chromatography? Well, chromatography doesn't necessarily have to do with just proteins, although it is certainly most commonly used to separate and purify proteins. So chromatography is just the process of separating molecules based on how they differentially interact with a solid medium. So basically what that means is that whatever your solid medium is, different kinds of molecules are going to interact with that medium in different ways, and chromatography takes advantage of that. The oldest and most simple type of chromatography is called paper chromatography, and this actually may be something that you did previously earlier in your education. So you can do something like this to separate molecules like ink or plant pigments, things that you do in elementary, middle school, or high school projects. That's why I said you may have done this before. So this is a good way to introduce ourselves to a concept that is common to all types of chromatography. You have two different phases that molecules can be a part of. So you have a stationary phase, which is going to represent our solid medium. In the case of paper chromatography, this is just going to be a piece of filter paper. So the filter paper in this case has different types of molecules blotted somewhere on it, usually towards the bottom. So if we have a mixture of molecules that we want to separate, we could just take that mixture and wipe it across the bottom of the filter paper. Now the molecules are still together at that point, but they become attached to the stationary phase. And the reason why we use the term stationary phase is because at this point, those molecules are not going anywhere. They are basically stationary. So for paper chromatography, we next will dip the bottom of the filter paper near where our molecules are into some type of solvent. This could be water, this could be alcohol, this could be some type of non-polar solvent. Doesn't really matter. We're going to expose the paper to some type of liquid solvent. The solvent is then going to migrate up through the paper, and this is why we call the solvent the mobile phase. So depending on what kind of molecules are in our mixture that we blotted onto the piece of paper, some of those molecules may really like interacting with the solvent, and they will transition from being a part of the stationary phase to being a part of the mobile phase. If this is the case, those molecules will travel up through the filter paper as the solvent migrates as well. But molecules that do not like the solvent are probably going to stay a part of the stationary phase, and they're not going to move as much. So as it says here, depending on how the molecule interacts with the solvent, their speed of migration will vary. So if you look at the initial point here where we take our mixture of molecules and just kind of swipe it across here so that we have one line, as the mobile solvent phase moves up through the paper, it carries with it 4 different molecules in the original sample, and they migrate differently based on how soluble they are in the solvent and in the mobile phase. So, like I said, this is the most simple and certainly the oldest type of chromatography that has been commonly used, but the general principles are essentially the same. Your ability to separate molecules by chromatography is going to depend on how those molecules interact differentially between the stationary phase and the mobile phase. So another very similar type of chromatography is called thin layer chromatography. Essentially, again, the same principles here. We have a mobile phase, which is some type of solvent. Again, it could be water or alcohol or something else. And we have a stationary phase, which instead of paper, we this time have a type of silica gel. So the same principles apply. Molecules in the sample, which are exposed to the silica gel towards the bottom of the plate, they will migrate through the gel as the solvent moves upward on the basis of how not only how large the molecules are, but how soluble they are in the solvent. For example, if water is our solvent, then a hydrophobic molecule like a fatty acid or an oil or something of that nature in the sample is not going to be carried very well in the mobile phase, and it's not going to migrate very far in the gel. But if our mixture of molecules contains something that's very hydrophilic and very much likes water, then it should travel fairly quickly and it should migrate further up into the gel. All right, so that is going to do it for this video. Just wanted to introduce some of the concepts and the terminology and some of the principles involved in chromatography in general, but for the purpose of purifying proteins, we're going to want our focus to be pretty much solely on column chromatography. So we're going to devote our next video to a discussion of the different types of column chromatography. So I'll look forward to seeing you next time, and that will be what we discuss then. See you later.